Hello everyone, I am Katerina. I recently completed my PhD working with fish health and nutrition. More specifically, my expertise lies on live foods, the ontogeny of the digestive system, and the ontogeny of the skeleton and skeletal deformities. And today I will present you some of the results of my PhD regarding the effect of using Acartia tonsa provided by CFEED on the development and ontogeny of Greater Amberjack and gilthead seabream. First of all, the Mediterranean aquaculture industry produces animal protein of high nutritional value, rich in highly unsaturated fatty acids. The most common commercial species in the aquaculture industry are the gilthead seabream, the sea bass, the red porgy, and the migr, while new species in the aquaculture industry are the Greater Amberjack and common dentex. In the present work, I studied Greater Amberjack and gilthead seabream. The domestication of new species, such as Greater Amberjack, faces several problems and difficulties, such as the lack of knowledge of the optimal environmental conditions and the lack of an established and tested feeding protocol for species in which the classical combination of rotifers and artemia is not sufficient. Live foods are the primary food source during larval rearing, and they are very important for growth and development. The movement of live organisms triggers the feeding reactions of the fish since they are visual feeders. Live prey is always accessible to the fish, unlike formulated diets which tend to sink to the bottom of the tanks. In addition, the digestive system of newly hatched larvae in most species is unable to process formulated diets. So, despite several advancements in the development of formulated diets, most species still rely on live foods. The most important live foods are microalgae, rotifers, artemia, and copypods. Rotifers have several advantages and thus they are widely used in the aquaculture industry. First of all, their body size can range between 70 and 350 micrometers. They have a high population growth rate and they are very tolerant to culture conditions. However, they are very poor in highly unsaturated fatty acids and thus they need enrichment. They have a high organic load, pathogenic bacteria, viruses, fungi, and they can be vectors of fish pathogens. Artemia, on the other hand, has greater body size, which can range from 350 to 500 micrometers for Artemia naupli and 700 to 800 micrometers for Artemia metanaupli. It has its unique ability to form dormant cysts, which are very resistant to environmental conditions, can remain viable for many years, and it is very easy to transport and store them. On the other hand, the cost of purchase is very high, and similar to rotifers, they are very poor in highly unsaturated fatty acids, and thus they need enrichment. Moving on to copypods, which were the live food that we tested in our experiments, their body size can range between 70 and 350 micrometers, and unlike rotifers and artemia, they are an important part of the natural diet of marine fish larvae, and have higher nutritional value than enriched rotifers and enriched artemia. So they do not need enrichment, and thus they don't burden the surface of the tanks with extra fat. Regarding the nutritional profile of copypods, they are very abundant in free amino acids, which can improve the growth and ontogeny, and they are also very abundant in highly unsaturated fatty acids in the phospholipids of their cell membranes. Thus, the highly unsaturated fatty acids in copypods are more available and digestible by the fish. Copypods also have lower bacterial load. There are some vitamins, such as vitamin C and vitamin E, which are very important for growth and vision. They can stimulate the immune system and stress resistance, and they are also very important for the normal development of the digestive system and the skeleton. In addition, some minerals, such as iron, selenium, zinc, copper, magnesium, and calcium, are important for the normal development of the skeleton and the digestive system, and more specifically, they can influence the proliferation of goblet cells in the intestine. Finally, fatty acids can increase survival rates after handling stress, increase growth rates, and they can also improve the ontogeny of the digestive system and the skeleton. In this table, we can see that the nutritional profile of copepods is much better than the nutritional profile of enriched rotifers, Artemia naupli, and enriched Artemia metanaupli. The first experiment was performed in a marine fish farm and was with Greater Amberjack. In this experiment, we used 4 cylindroconical tanks of total length of 2,700 liters each, and we stocked each tank with 55 larvae per liter. We used 2 tanks for the control group and 2 tanks for the copepods group. The feeding protocol was the same for both the groups, the control and the copepods group, and the only difference was that the fish from the copepods group were fed additionally with copepods from the initiation of the exogenous feeding on day 3 after hatching until day 17 after hatching. Throughout the whole experiment, we conducted regularly samplings in order to study the ontogeny of the digestive system, the ontogeny of the skeleton, and almost by the end of the trial, we conducted one sampling in order to study skeletal deformities. The second trial was performed at the University of Patras in the Department of Biology and was with seabream. For this trial, we used cylindroconical tanks with total volume of 100 liters each, and we stocked each tank with 100 larvae per liter. For the first seabream trial, we used 2 tanks for the control group and 2 tanks for the copepods group. The feeding protocol once again was the same for both the control and the copepods group, and the only difference was that the fish from the copepods group were additionally fed with copepods from the start of the exogenous feeding until the end of the trial on 17 days after hatching. In addition, in this experiment, we use live microalgae in order to apply the Greek water technique and perform samplings regularly in order to study the ontogeny of the digestive system. Since this trial lasted only 17 days after hatching, the study for the ontogeny of the skeleton was not performed. And finally, a second seabream trial also conducted in the Department of Biology at the University of Patras. In this trial, we used 6 cylindroconical tanks, 3 tanks for the control group and 3 tanks for the copepods group. Once again, the feeding protocol was the same, and the only difference was that the fish from the copepods group were fed additionally with copepods from the start of the exogenous feeding on 3 days after hatching until 17 days after hatching. And once again, we used live microalgae in order to apply the green water technique and perform samplings regularly in order to study the ontogeny of the digestive system, the ontogeny of the skeleton, and by the end of this trial, we performed one sampling in order to study skeletal deformities. First of all, I would like to mention that both species, both Greater Amberjack and seabream, indeed consume copepods. We can see in both pictures with the red arrow to indicate the eggs of rotifers, while the black arrows indicate the residual of copepods naupli. It is very important to mention that the fish larvae of Greater Amberjack that we used to test if the larvae eat copepods had never fed with copepods before, but they eat them nevertheless. Moving on to our main experiments, we can see that in Greater Amberjack, the mortality rate was higher in the fish from the control group than in the fish from the copepods group, especially from days 10 until 17 after hatching. On the other hand, there were no statistically significant differences in the mortality rates in both seabream trials. Regarding the growth of the fish, we can see that in both Greater Amberjack and the fish from the second seabream trial, in both those species, the use of copepods affected positively the growth of the fish, especially during the last samplings. Moving on to the ontogeny of the digestive system. In these tables, we can see the ontogeny of the digestive system in both Greater Amberjack and seabream. Regarding the Greater Amberjack, we can see that pyloric cica appeared earlier in the fish from the copepods group than the fish from the control group. This practically means that the copepod-fed fish had extra area for digestion and absorption of nutrients. Regarding the seabream, we can see that the goblet cells in the esophagus appeared earlier in the fish from the copepods group. In addition, the goblet cells in the intestine appeared in the fish from all the 3 tanks of the copepods group by the end of the trial, but only in one of the tanks from the control group. This practically means that the copepod-fed fish had more goblet cells and thus more mucus. Regarding the length of the villi, we can observe that in both Greater Amberjack and the second seabream trial, the copepod-fed fish had higher length of the villi. So, in both species, the use of copepods affected positively the length of the villi, which means that the copepod-fed fish had extra area for digestion and absorption of nutrients. We can see the same results regarding the surface of the villi. Once again, in both species, the use of copepods affected positively the surface of the villi, which again means that the copepod-fed fish had extra area for digestion and absorption of nutrients. Moving on to the abundance of goblet cells in the anterior intestine, we can see that in both species, the use of copepods affected positively the abundance of goblet cells, which practically means that the copepod-fed fish had extra secreted mucus. But what does this mean for the fish? So the copepod-fed fish with extra mucus had a better lubrication in the digestive tract, which means that there was a prevention of mechanical injuries. Mucus also increases the defense mechanism against pathogens, protects the intestinal segment from digestive enzymes that might cause autolysis, contributes positively to digestion processes, impacts osmoregulation, and contains a variety of ions which control protein transportation. In these histological sections, we can see the gradual ontogeny of the intestine in Greater Amberjack in both the control and the copepods group. The black circles indicate the goblet cells, while we can see that only in the fish from the control group there were signs of desquamation. Here we can see the gradual ontogeny of the intestine in the seabream, once again from both the control and the copepods group. And similar to Greater Amberjack, we can see that only in the fish from the control group there were signs of desquamation. Moving on to the liver, we can see in both species the use of copepods affected positively the fat accumulation in the liver, which means that the copepod-fed fish had extra energy for growth and development. However, it is very important to mention that in both species during the last samplings, there was a reduction of the fat accumulation in the liver, especially in the fish from the copepods group. This reduction means that the fat in the liver was used for body growth. And actually we can confirm that because during the same samplings, the fish from the copepods group was bigger than the fish from the control group. In these histological sections, we can see the gradual ontogeny of the liver in both the control and the copepods group in Greater Amberjack. And we can see that especially in the fish from the control group, there were significant signs of hyperemia. And here we have the gradual ontogeny of the liver in seabream. The results for the first seabream trial showed us that the results were similar. Once again, the length and surface of the villi and the area covered with lipid vacuoles in the liver were higher in the fish from the copepods group. The normalization of the data from the ontogeny of the digestive system showed us that in both species, the length of the villi, the abundance of goblet cells in the intestine, and the area covered with lipid vacuoles in the liver were affected directly by the diet, the use or not of copepods. However, the surface of the villi was affected by the growth of the fish. But how come the surface of the villi was affected by growth, but the length of the same structure was affected by the diet? The answer lies to the growth rate. More specifically, we can see that the total length of the fish and the surface of the villi exhibited an exponential growth rate, which means that when there was a peak in the total length of the fish, there was also an increase in the surface of the villi. On the other hand, the total length of the fish and the length of the villi did not show an exponential growth rate, which means that when there was a peak in the total length of the fish, there was not necessarily an increase in the length of the villi. So the body length increases in parallel with the surface of the villi, but not with the length of the villi, which was exclusively affected by the diet. So the copepod-fed larvae had better ontogeny of the digestive system. But why? The answer lies to the nutritional profile of copepods, which is better than the nutritional profile of enriched rotifers and enriched artemia. More specifically, fatty acids, which are more plentiful and easily absorbed were ingested from copepod phospholipids rather than enriched products, can enhance the activity of the digestive enzymes and increase the area covered with lipid vacuoles in the liver. In addition, there are some micronutrients such as iron, zinc, selenium, copper, magnesium, calcium, and vitamin E, which are more abundant in copepods and can be very beneficial for the ontogeny of the intestine, and more specifically, the proliferation of goblet cells. Moving on to the ontogeny of the skeleton. We can see that in both species, the structures appeared earlier in the copepod-fed fish. More specifically, in Greater Amberjack, those structures were the neural process, the predorsal, and the proximal pterygophore in the dorsal fin, and in the seabream, the vertebral center, the proximal pterygophore in the anal fin, and the hypural. In addition, we can see that the copepod-fed fish had completed the formation of the majority of the structures earlier than the fish from the control group. Those structures in Greater Amberjack were the predorsals, the proximal pterygophore of the dorsal and the anal fin, and the lepidotrichia in the pelvic fins, and in seabream, the proximal pterygophores in the dorsal, the anal, and the pectoral fins. Now, regarding the calcification, we can see that in Greater Amberjack, the majority of the structures that we study completed their calcification earlier in the fish from the copepods group. And these structures were the hypurals, the vertebral centers, the neural processes, the hemal processes, and the proximal pterygophores of the pectoral fins. On the other hand, only in the copepods group in seabream trial, the clithrum in the pectoral fins was fully calcified. Now, regarding skeletal deformities, we can see that in Greater Amberjack, more than half of the fish from the control group had at least one deformity, while this percentage in the fish from the copepods group was not more than 40%. More specifically, we can see that the fish from the control group had severe deformities in the vertebral column and the caudal fin, while only in the fish from the copepods group we were able to find deformities in the pectoral fins. The deformities in the vertebral column and the caudal fin are very important because they are often fatal to the fish since they impair their ability to swim quickly in order to feed. Regarding the second seabream trial, half of the fish from the control group had at least one deformity, while this percentage in the fish from the copepods group was quite low. More specifically, there was an increased appearance of scoliosis in the control group, while only in the fish from the copypods group we were able to observe deformities such as broken hemal processes and fusion of the vertebral centers. Excuse me. Okay. Here we have some pictures of skeletal deformities in both Greater Amberjack and seabream. More specifically, we have a deformity in the 23rd neural processes and the epiureals, in the first hemal processes, in the metaclithrum lower in pectoral fins, the second proximal pterygiophore in the anal fin, the fourth proximal pterygiophore in the dorsal fin, the predorsal, and a case of crossbite. While in seabream we have a fish with kyphosis, lordosis, scoliosis, fusion of the vertebral centers, broken neural processes, and broken hemal processes. Now, regarding the inflation of the swim bladder, there were no statistically significant differences between the fish from the control and the copypods group in the Greater Amberjack trial. However, in the seabream trial, we can see that there were significant differences between the two feeding protocols regarding the inflation of the swim bladder. In addition, 9% of the fish from the control group had a hyperinflated swim bladder. But can this be a result of the diet? The answer is yes, since the copypods do not need enrichment and thus they don't burden the surface of the tanks with extra fat, and the fish have an uninterrupted and better inflation of their swim bladder. But regarding the seabream trial, can the swim bladder anomalies affect the skeletal deformities in the vertebral column? Let's see first the case of scoliosis. We can see that the fish with scoliosis was much more in the control group than in the copypods group. The first fish with scoliosis were observed very early on day 7 after hatching, which is too early for an inflated swim bladder. In addition, 82% of the fish with scoliosis had this particular deformity in the caudal region, which cannot be affected by the appearance of or not of an inflated swim bladder. So the answer is no. However, the cases of scoliosis on day 7 are in the notochord and not in the vertebral column. Deformities in the notochord are often parental or a result of environmental conditions. But since the fish from the copypods group had less cases of scoliosis by the end of the trial, this could mean that the use of copypods maybe had restored the scoliosis in the notochord. Of course, there is a need of additional trials in order to confirm this hypothesis. Moving on to the cases of lordosis and kyphosis, we can see that the number of fish with lordosis and kyphosis in the fish from the copypods group are almost similar with the fish from the control group. So once again, the swim bladder anomalies cannot have affected the skeletal deformities in the vertebral column. In order to test whether the differences in the ontogeny of the skeleton was affected directly by the diet or the growth of the fish, we calculate the growth rate between the sampling points in both species, and we saw that there were no statistically significant differences between the growth rate between the two different feeding protocols. So, any differences in the ontogeny of the skeleton can be attributed directly to the use or not of copypods. So, the copypod-fed larvae had better ontogeny of the skeleton and less deformities. The answer to this lies once again to the nutritional profile of copypods. More specifically, fatty acids can cause to a faster skeletal ontogeny. When in the foods there are more phospholipids, they have been recording less skeletal deformities, while when in the foods there are less free amino acids, we have more skeletal deformities in the fish. In addition, the mean optimum DHA-EPA ratio has been calculated between 2.2 and 2.4, while enriched rotifers have this ratio in 2.8, enriched Artemia 1.4, and copypods 1.8. When the DHA-EPA ratio is close to 2 and in copypods is 1.8, this is related to the expression of some genes which are related to the better and earlier ontogeny of the skeleton, earlier calcification, and less skeletal deformities. In addition, there are some macronutrients such as iron, iodine, selenium, copper, magnesium, and calcium, which are more abundant in copypods and can be very beneficial for the ontogeny of the skeleton. In order to summarize, the use of copypods Acartia tonsa during the first days of rearing of Greater Amberjack at seabream had a positive effect in growth and development and the ontogeny of the digestive system. More specifically, the fish from the copypods group had higher length and surface of the villi, increased number of goblet cells in the intestine, higher percentages of fat accumulation in the liver, and in the case of Greater Amberjack, earlier appearances of the pyloric cica. This practically means that the fish from the copypods group had larger area for digesting food and absorption nutrients, easier digestion, more efficient absorption of nutrients, amplification of the immune system due to the mucus, and extra energy. In addition, the copypod-fed fish had a better ontogeny of the skeleton. More specifically, the copypod-fed fish had earlier first appearance of structures, earlier completion of formation of some structures, and earlier completion of calcification. Had also less skeletal deformities, and in case of seabream, more successful inflation of their swim bladder. Thank you very much for your attention.
Thank you. It was really good to see the results, and I think it's very interesting, the findings on the digestive systems. Do you think that these findings can be linked to the improved FCR that we see?
Yes, because since they have the ability to digest more food, that could affect the FCR.
Thank you. Is there any other questions for Katerina? Yes, Sami? Let me see, you're muted.
Yeah, okay. You hear me? Yeah.
Yep.
Thank you very much for these nice results. Going in detail in the digestive system is, not very common and very good. I have just two small questions. When you add the co-feeding copypods, so means that you did not show us, but it's not the concentration you are using. And if you add copypods, so you add globally the quantity of mass that you use it. You did not try to give them the same amount of equivalent carbon or nitrogen or equivalent protein. So because you added the quality is copypod, but you added a few the quantity also. So can you comment why you use, because you give double privilege, more quantity and more quality, because the copypod are superfood for the larvae. So
okay, actually the quantity didn't change because we tried to have a stable quantity of carbon in the tanks. For example, in the seabream trials, I cannot say much things about the trial of Greater Amberjack because I did not participate in that. But regarding the seabream trials, in the fish from the control group, in the tanks from the control group, we added 5 individual per ml, while in the tanks from the copypods group, we added around 2.5 rotifers per ml and 2.5 copypods per ml. So we tried to keep the quantity the same as much as possible, but of course to have a different quality of the food.
Okay, because it was not clear from the protocol. Now it's clear. And the second question, in your control of seabream, you reached 50% of malformation. Is it this number common in industry hatchery, or is it quite high? You have 50% of, deformed and 17 point something in copypot, which is a good result. But did your control 50% is not too high compared what we know from literature and maybe in hatcheries?
Yeah, in an industrial level, this percent it's very high. And I don't think that the majority of the hatcheries have such a high percentage of deformities, especially if we observe that the most common deformity is scoliosis. However, the deformities have to do with genetic reasons, environmental conditions, and we had the quality of the eggs was the same, and the environmental conditions for both the control and the copypods group were stable. So the only difference was the diet. I cannot answer you directly what happened with the control fish and had so many deformities, because especially if I can correlate this with the appearance of deformities in the notochord, which means that has nothing to do with the diet, but maybe with the genetic factors, and since environment was the same. But we can see that whatever happened or whatever the quality of the eggs, the use of copypods can, let's say, fix so many things. And as I comment, we cannot say this for sure, but just an hypothesis or maybe an idea for in order to add more trials and experiments. If it is a possibility that the use of copypods can restore some deformities.
Okay, thank you very much.
Thank you.
Yeah, thank you, Sami. Raya, you also have a question?
Yes, it's nice to know how potential our product is. So I have 2 or 3 queries, not questions, basically, about the during different feeding protocols. Do you see, Katerina, any difference of the gastric emptying process? Is any feeding protocol has a better gastric emptying process? That is number 1. And the second question is, did you have any data or observations on the intestinal health when they grow up to bigger ones which are fed with copypods?
So first of all, thank you for your questions. I'm afraid you have to repeat the first question because there was a pause in the internet and I didn't hear it. But regarding the second question of the intestinal health, we did not. I did not perform an extensive health test in the intestine of the fish. However, in both the Greater Amberjack and the second seabream trial, through histology, which I performed histology in order to see the ontogeny of the digestive system, there were signs of desquamation, not important, something very important that could be fatal to the fish, but this signs was observed only in the fish from the control group.
Good. Now, the first question was about the gastric emptying. The gastric emptying process.
No, we didn't do something. We didn't test something like that.
Okay. Good.
Super. Thank you. Does
Question?
anyone else has a question for Katerina? Maria.
Hello. and thank
Hi.
you for a very nice presentation. And congratulations with your PhD. It was some very nice results, and just wonder about the survival. I'm not sure if you mentioned it. Was it? Yeah, there it is.
We had
So
yeah, please.
Yeah, no, I was just wondering if you see this when you see scoliosis this early. I'm just wondering if they are dying early, that they kind of go out of the.
In cases of severe scoliosis, I believe that the fish will die at some point. However, since the sampling for skeletal deformities was performed on day 25, and we found fish with scoliosis, this means that the fish survive with scoliosis. But these fish survive with scoliosis in very small tanks of 100 liters with continuous feeding. I'm not so sure that they would survive in an industrial level. I will not say anything about the nature. They will not survive, maybe not until day 10. But an important factor is the shape and size of the tanks. We can see some things in small experimental tanks like the ones I used, which had a total volume of 100 liters. And but this is not. It is not necessary that we will see the same results, especially regarding deformities, inflation of the swim bladder, and mortality rates in bigger industrial tanks. And it has to do with the volume of the water and the density of the larvae inside the tanks.
Can I have one more question?
Yes, of
Okay.
course.
Katerina, you showed the slide about the energy, increase in energy because of the surface area, the increase of surface area of the digestive system, right? Are you talking about the energy intake or the metabolic energy there?
The energy intake. I cannot say something about the metabolic energy because I don't have run a test to prove something about how much of the food intake they metabolized and how much of this food intake they used for growth or for other aspects of their ontogeny.
So it's kind of compensating the energy requirements at that particular phase of growth.
Yes.